so much for coming to this talk. Really privileged to be a part of this wonderful session with many great speakers, and really privileged actually to follow Professor Kripps's opening lecture on causation in healthcare. So the title of my talk talks about my experiences as an applied biostatistician. So I wanted to give some context for where I'm coming from, where my perspective comes from. My basic training, my focus trainings in epidemiology, biostatistics, and statistical genetics. But I also have a lot of complementary training informing my understanding in econometrics, ethics, public health practice and policy, and so on and so forth. They've been very valuable in trying to understand what we can do with data and how people know what they know. So in my position at the Singapore Institute for Clinical Sciences, I lead studies on maternal and child health and generally how to improve human health and potential, with some specific foci in genetics, environmental causes, and methods development. But I think the most important part of what I want to talk about this evening is my work in consulting with clinician scientists on a number of different projects and domains, from clinical trials and nutrition studies to building clinical prediction models in a number of domains, metabolism, mental health, and dentistry. And in this work, I often hear, and oftentimes you will hear, let's just see what the data say, or what insights can the data give us? We might be shown reports that are prefaced by the data show or these data prove. But really, and this is something you've probably heard before, data don't speak for themselves. In much the way the ventriloquist dummy doesn't speak for itself, really the use of data and the presentation of data and the interpretation of data comes from the analyst, comes from us. And I think it's really important to understand that. But I think that that's something that people understand very well. I want to take this a bit further and maybe be a bit deliberately controversial. And it might seem strange given that my life and work revolves around data. But I think that when it comes to data analysis, the solution does not sit in the data. So what do I mean by that? So consider the following results from a particular study. After 6 months of follow-up, a group given treatment X has an average of a 5.5 millimeters mercury reduction in blood pressure, and that there was no change in the group receiving no treatment. How do we interpret that? Well, in order to give those results any meanings, even if they might be completely correct, is we need to be able to answer other questions. Were individuals randomized into the treatment? Was a set of assignments sufficiently randomized and blinded? Was there substantial crossover in treatments? Was there good compliance? And generally speaking, the answer to those questions don't sit in the data. And so it's impossible to really answer the questions and therefore interpret our findings without outside knowledge, whether it be about the study design, what are important other factors, really without general expert knowledge. And this relates very much to what Professor Kripps said before about incorporating domain expertise. It also leads to the principle that the data by themselves don't necessarily inform how you should analyze them. So I want to explore this a bit further, but just to give a toy example about how this might arise. So consider you're trying to estimate the effect of a treatment on some X on some response Y. You're given some raw data from the client that's plotted here. Along the X-axis is the treatment, and the Y-axis is the response. In this shading, this colored shading is some covariate W, and there's many other covariates. So what do? You apply your favorite methods to generate a response model for the treatment on the response, using your best ensemble learning, machine learning approaches with a lot of cross-validation, hyperparameter tuning, and you see this particularly interesting finding result. And also you're heartened by the fact that whatever fit measure you use, say a R squared or a pseudo R squared, is 0.4. So your model seems to do a fairly good job of explaining your data. But hold on, actually in reality, the values of your covariate W that you included in your model are actually something that are influenced by treatment X and response Y, and something that you would have found out if you'd done the directed acyclic graphic exercise, probably as advised by the previous speaker. Say this is some factor such as a length of hospital stay that's influenced by both the treatment and response, but not a direct causal element. And in fact, if you properly model this and you didn't include covariate W, you see that the true effect is linear. But interestingly, the amount of variance explained by this particular model is 0.2. It's less than the best fitting model that you have provided before. And that's really the heart of the challenge of trying to understand effects from data, is you are only interested in certain portions of the variance, the relevant variation that's due to causes of the Y, including the treatment that you're studying. And this is not immediately verifiable by any internal prediction metric or correlation. And it's not an overfitting issue that people who are familiar with prediction models are familiar with, in that this is not an issue of one particular sample of the data, but this is an issue that will arise if you model the data in the same way in any data set in any setting and continually get to what is the incorrect answer or biased answer. And the only solution to this is to have outside knowledge. But there's upsides to this, because when we start thinking about how we use our outside knowledge, it leads us to good questions that'll improve the analysis and the findings from our studies. And so we can ask questions like, what relevant outside knowledge will allow me to design a better analysis given the available data? So to give one example from the work that we've been doing, we were interested in looking at the effects of in vitro fertilization or assisted reproduction on child health and development. And to sum up our results, we find what several other studies and some meta-analyses have found, which is that assisted reproduction is related to smaller and shorter children. And the question, of course, is how. And knowing how will help us figure out how to intervene, or whether or not we can possibly intervene. One potential mechanism by which this is hypothesized to happen is through early developmental changes, specifically epigenetics, but we don't need to get into the specifics of that. It's just important to know that some exposures, for example, assisted reproduction that happen early in life, can affect biological function in a persistent manner. And so that's all well and good, but unfortunately, these particular kinds of changes, these epigenetic changes, may also be due to other factors, and particularly problematically, unmeasured factors such as maternal or paternal health and diet, things that don't exist in our data, but are very important. Also important for this context and in many other health research contexts, we can't do randomized experiments for a number of reasons. One of them is because assisted reproduction is actually very effective. So denying it is not sort of an ethical choice. The other one is the sort of cost and difficulty of following up trial patients for an extended period of time and being able to collect all the information that we want to. And thinking about applying our machine learning approaches, we can't learn from data that aren't there. At least I don't think that we can learn from data that doesn't occur in our data set. So the missing data on maternal health and diet, for example. So how does health expert knowledge help us inform a solution? Well, one way is to think about how we would approach this if we were able to do an experiment, and try to figure out, using the data, whether or not we can do similar types of experiments. And one method that's well known to experimental scientists is the approach of using positive and negative controls. So a negative control in an experiment might be something where you haven't included the active ingredient, so you know the result that's supposed to happen, which is that you're not supposed to get any reaction or anything showing up on the plate. So if we incorporate outside knowledge, there are ways to think about how we could do this in observational data and the data that we've collected. So for example, if we wanted to look at the effect of in vitro fertilization on the developing baby through its epigenetic changes, ultimately on its growth and development, one idea is to use the epigenetic changes or epigenetic differences in the mother as a negative control, because these may affect the developing baby, but it shouldn't affect directly this pathway that we're interested in. And in fact, not only can it be a negative control, but we can use the information about how this association that we posit not to exist as a proxy for this unmeasured confounding that we have. And to sum things up, we want to do some simulation studies to reassure us of the fact that it's actually possible to do these kinds of things. So we set up a simulation in which the maternal epigenome proxies for some of this unmeasured confounding. And what we see in the results here on the left is if we don't do anything to take into account the unmeasured confounding, it's what we expect. The effect is biased in that these bars are away from the true effect shown by the dotted line. But when we take into account this unmeasured confounding with this negative control, with this proxy, we can recover an effect. So we showed this in our study. We were able to show, for example, that this particular gene, methylation, was related to the, was mediating part of the effect of in vitro fertilization on child height. So then, say we have our design and we're happy with it, we have enough external information. The other question is, how do I ensure that the machine learning I'm using is appropriately leveraged for that particular data context? In general, what we want to do very simply is to estimate the effect of some binary treatment A on outcome Y, something that we commonly do. We try to estimate an average treatment effect. And we often do that through the use of doubly robust methods, which involve estimating a treatment model or propensity score and an outcome model, which is something that people are usually fairly familiar with, for example, a conditional regression model. And it's considered doubly robust in a way that a belt and suspenders might be considered to be doubly robust, in that either one of them can function to hold your pants up or to protect against bias, but together they can work even better. So one of the major benefits of using these approaches is that they are flexible and they allow the use of your favorite machine learning techniques. For example, weighted meta-learners of cross-validated algorithms. They are relatively efficient in smaller samples, which is something that's very important for clinical and biomedical studies that often are very expensive to do and can only be done in smaller samples. The cons are that they are computationally expensive. They may actually be biased in smaller samples when the particular data setting and the data support is not quite right. And it's not quite clear how much misspecification these approaches actually protect against. And it really depends on the specific setting of the data. And it's quite difficult to understand. Again, it doesn't come from the data itself when these concerns are applied, when we can use these methods or not. So we set out to do some simulation studies. Do machine learning methods work well for small biomedical cohort data when using flexible estimators? In general, when the models are correctly specified, they generally don't outperform more simple types of regressions when using these estimators. However, in the case where there are many excess covariates, or when the covariates are not that important to the model, machine learning methods can have a benefit, but only when fit in a particular way that is fairly computationally expensive. So we've developed a tool that should be available fairly soon for people to be able to put in their own data to evaluate whether or not these particular methods are valuable or unbiased in their particular setting. And these kinds of applied approaches are really essential to be able to make use of these machine learning algorithms. So I've just briefly gone over two examples of what we do and how we think about causation in healthcare. I think the general upshot is that we're not limited by data or computational power these days. Subject matter experts really look to us to analyze, interpret, and inform actions. But this can't be done without a high-level understanding of both how the data context, and that includes all the background science and the use case, the relevant biology, integrates with your particular analytic design. And there's no replacement for it. And this comes from interactions with a lot of these domain experts who really look to you to inform on both sides of these analyses. So with that, I'll end. Thanks to funders, some excellent students I've worked with, and you for listening.
Thank you, Jonathan, for your interesting and insightful sharing. I can see we already have a couple of questions coming up. The first question is data quantity or data variety more important? Jonathan, do you want to take that?
Data quantity or variety? I would think, the correct answer is that the right data is what you need. So this is an open-ended answer that it has to be the specific kind of data that will answer your question. Now, once you have that, then there's a question of whether or not you have enough data. But I think it's much more important to figure out whether or not you have the right data first. And part of understanding the right data is something that I mentioned at the beginning, which is you have to understand where the data comes from, right? Does it come from a trial? Does it come from a very select population? Very early on, for example, in our COVID pandemic, we had so much research, but on data that was collected very haphazardly. You don't get good results. No matter what kind of data you collect, how it's collected is really important as well.
Makes a lot of sense. We have another question, which is more technical. The question is, can you account for the missing data where you can't run an randomized control trial through latent variable modeling? Do you want to answer that, Jonathan?
I think that you can if you're confident that your latent variables capture actually the sources of confounding, and that's not a simple effort. Even if you have, for example, the right variables, if they're not parameterized in the right way, if you don't have the right model, and this is something that machine learning is actually very good for, is if you have all of the right models, machine learning can help you search through the right models that will create the best fit for a particular objective. But the challenge is, of course, defining that objective and having the right variables. But yeah, they can.
Thank you, Jonathan. Okay, thanks again for your sharing. And now let me welcome our next speaker, Professor Wilson Goh from Nanyang Technological University. Over to you, Wilson.